Good day everyone, I am Jana P. Aguilar and today I am going to discuss to you the basic concepts of human reproduction. So first I am going to define what is human reproduction. So human reproduction is any form of sexual reproduction resulting in human fertilization. It typically involves sexual intercourse between a sexually mature male and female. So, during sexual intercourse, the interaction between the male and female reproductive system result in fertilization of the woman's ovum by the man's sperm. So, these are specialized reproductive cells called gametes. It created in a process called meiosis. While normal cells contain 46 chromosomes, 23 pairs, gamete cells only contain 23 chromosomes. And it is when these two cells merge into one zygote cell that genetic recombinations occur in the new zygote contains 23 chromosomes from each parent, giving them 23 pairs. So a typical 9-month gestation period is followed by childbirth. The fertilization of the ovum may be achieved by artificial insemination methods which do not involve sexual intercourse. Assisted reproductive technology also exists. So now we are going to proceed to heredity. So when we say heredity, it is the mechanism for the transmission of human characteristic from one generation to the next. So each person carries a genetic code or genome, a complete set of code instruction for making and maintaining an organism. The genome is inherited from both parents. So when we say genome, it is described as the blueprint or book of human life. It carries and determines all the characteristics of a person yet to be born. The genome is located within each of 100 trillion cells in the human body. The nucleus inside the cell contains a complete set of the body's genome that is twisted into 46 packets of thread-like microscopic structure called chromosome. So when we say chromosomes, it is the genetic material present in all cells. So the chromosomes comes in 23 pairs. So each pair is composed of one chromosome from the male and one chromosome from female or parents respectively. Each set has 22 single chromosomes called autosomes that carry the physical, mental, and emotional or personality characteristics. So when we say autosome, it is a chromosome in a eukaryotic cell that is not a sex chromosome. So each autosome stores many thousand genes, each of which performs a unique function in the organism cell. So when an autosomes are healthy, this enables cells to perform an awesome array of functions. Each of hundreds of subtly differing cell types in a eukaryotic organism express a different combination of genes in the right place at the right time, enabling the huge array of cellular functions we see in eukaryotic organisms like ourselves. A female will have a pair of XX chromosomes while a normal male will have an XY pair of chromosomes. So inside the chromosomes is the long thread-like molecule and genetic substance called the deoxyribonucleic acid or DNA. So when we say DNA or deoxyribonucleic acid, it is the hereditary materials in human and almost all organisms. Nearly every cell in a person's body has the same DNA. So most DNA is located in the cell nucleus where it is called nuclear DNA. But a small amount of DNA can also be found in the mitochondria where it is called mitochondrial DNA or empty DNA. So mitochondria are structures within cells that convert the energy from food into a form that cell can use. The information in DNA is stored as a code made up of 4 chemical bases, which are adenine, guanine, cytosine, and thymine. So adenine bases located on one strand form chemical bonds with thymine bases on the opposite strand, and the guanine bases located on one strand from chemical bands with cytosine bases on the opposite strand. Next is cytosine. So cytosine bases located on one strand from chemical bands with guanine bases on the opposite strand. And lastly, we have thymine. So thymine bases located on one strand form chemical bands with adenine bases on the opposite strand. So now we will going to proceed to some principles of genetic determination. So when we say genetic determination, it is a complex affair. Much is unknown about the way genes work, but a number of genetic principles have been discovered. Among them, the principle of dominant recessive genes, sex-linked genes, polygenically inherited characteristic, and reaction range and canalization. So first, we have dominant recessive gene principle. If one gene of the pair is dominant and one is recessive, the dominant gene exerts its effect, overriding the potential influence of the recessive gene. For example, brown eyes, farsightedness, and dimples are common dominant genes that rule over blue eyes, nearsightedness, and freckles. A recessive gene exerts its influence only if the genes of a pair are both recessive. If a recessive gene is inherited from only one parent, the trait will not show. The person may never know that he or she carries the recessive genes. The second one, we have sex-linked genes principle. These are the genes that are in the sex chromosomes and that are therefore inherited differently between males and females. As mentioned earlier, females have two X sex chromosomes and males have an X and a Y sex chromosomes in their respective karyotypes. When one X female chromosomes combine with the X male chromosomes, the XX chromosomes result that make the organism a female. Meanwhile, when one female X chromosomes combine with the Y male chromosomes, the XY combines result that makes an organism a male. And the third one is the polygenic inheritance principle. So genetic transmission is usually more complex than the simple example mentioned earlier. So when we say poly, it means many. Genic means genes. So inheritance describes the interaction of many genes to produce a particular characteristic. Considering that there are as many 140,000 genes, the huge number of combinations possible is hard to imagine. Traits that are produced by the mixing of genes are said to be polygenically determined. So lastly, we have genotype and phenotype genetic heritage. So nobody possesses all the characteristics that the genetic structures make possible. So when we say genotype, it refers to the person's genetic heritage or the actual genetic material. So the genotype is established at conception during the process of fertilization and usually remains constant and does not change. So on rare occasions, the constancy is disturbed when mutation takes place or errors in cell division alter subsequent cell division. So genotype is not readily available for actual inspection. When we say phenotype, it refers to the person's observable traits that may be used to draw inferences about the genotype. So, the phenotype is the observable result between the genotype and the environment. That will be all. Thank you for listening.